Good evening and welcome once again. This is part 4 of Staphylococcus aureus, the discussion that we are going on. We are discussing, and we have already done the etiology, the pathogenesis, and the epidemiology. So at this moment, we are at the diagnostic strategies. So let's continue. The early and accurate diagnosis of Staphylococcus aureus is crucial in this, as it eventually affects the approach to treatment options. The gold standard remains culture-based methods. This involves collecting a sample which is then grown on a sheep blood agar plate for primary isolation. The bacterial colonies isolated are evidenced by complete lysis of the red blood cells by the beta hemolysis, forming a clear zone around the bacteria. A coagulase test is then performed to distinguish Staphylococcus aureus from other coagulase-negative Staphylococcus, and this takes around 48 hours turnaround, making the method less considered due to treatment delays. The other method is the use of molecular techniques such as real-time polymerized chain reaction, and this is a technique that uses amplifying the MEK-A gene to confirm the MRSS strains, and it is highly sensitive and specific and has a shorter turnaround of 3 to 6 hours compared to the previous. Lastly, we have rapid diagnostic tests that such as matrix-assisted laser desorption ionization, the MALDI-TOF-MS, the technique that uses laser ionization of bacterial proteins which are separated and detected based on their mass to charge ratio using the time of flight TOF mass spectrometer. Another method that is occasionally used is latex agglutination, which detects protein A, a protein found on the cell wall of the Staphylococcus aureus that binds to fibrinogen in plasma, causing the bacteria to clump together when exposed to antibodies specific for the fragment crystallizable Fc region of the IgG. It's simple and easy, but not as sensitive as the PCR. Although these methods have been commonly used, the biofilm formation by Staphylococcus aureus shields its detection, and this may result to false cultures. The PCR has also a challenge of detecting MEK-A gene, but cannot confirm viability, which can lead to overtreatment, and care has to be done to make sure that the diagnosis is really accurate.